All right, hey everybody, welcome back to our next video. This one is going to be on muscles. So we're going to be talking about the muscle system and seeing the specifics of muscles of the human body. So here we go. Muscles of the human body. This is going to be a generalized overview. So this is going to be a lecture. We're going to have major functions aligned with the musculature of the body. What are those major functions? Well, the major functions are going to be to move the body, maintain bodily posture, protection and support, regulating the elimination of materials, such as with the digestive tract, and heat. Each time we contract our musculature, we give a little bit of heat as it goes off. So we have a little bit of a matching game here. So for bodily movement, which one of these letters are we going to have? Well, first of all, we can see that E is the correct choice. Move the bones, make facial expressions, speak and breathe. All right, maintaining of posture. What does that mean? Well, C is the correct choice there, stabilizing joints and maintaining body position. Protection and support. All right, packaging of the internal organs and holding them into place. Regulation of elimination. That would be D, the circular sphincters controlling the material passage through orifices. And our heat production is going to be helping to maintain body temperature. So we have 5 characteristics of muscle cells, the cells that make up musculature. They are excitable, meaning that they respond to electrical change. They are conductive, meaning that they can conduct electrical currents. They are contractile, meaning that they are able to contract. They're able to pull on each other. They are elastic, meaning that they can stretch and they can move and they are malleable. And extensibility, right? So let's go ahead and match these with what we've got going on here. Excitability, that is going to be A, the ability to respond to a stimulus, right? We need to go all the way back there. Conductivity, right? This is the ability to send an electrical signal down the length of the cell membrane. So D would be the correct response there. Contractility, that would be C, filaments sliding past each other, enabling the muscle to cause movement. Elasticity, again, this is going to be the ability to return to the original length following a lengthening or shortening. And extensibility is the ability to be stretched. So we have 3 layers of anatomy here for a muscle. On the smallest degree, we have a muscle fiber. This is an individual muscle fibril or a myofibril. Each myofibril is going to be packaged into a larger group of myofibrils known as a fascicle. And a muscle fascicle composes several different myofibrils. Each fascicle is then packaged into a unit that is known as the muscle as a whole. So we have 3 levels of organization. We have a whole muscle, we have each smaller fascicle unit of the muscle, and then we have small myofibrils inside of there. So a muscle fiber, the individual myofibril is going to be covered by connective tissue known as endomysium. Endo meaning inside, mysium meaning muscle tissue. So it is covered by areolar connective tissue known as the endomysium. The fascicle is going to be covered by what is known as the perimysium. Peri in the middle, this is a mid-level structure. So this is dense irregular connective tissue that is going to be wrapping around the fascicle. And then the muscle as a whole is covered by our epimysium. Epi meaning above or outside. So the epimysium is a dense irregular connective tissue sheath that is going to be wrapping around the entirety of the epimysium. It's going to be heavily innervated, so we're going to have a lot of blood flow. We mainly see nerves and blood vessels flowing through the perimysium and the deep fascia of the muscle tissue. Each muscle is attached to something else by a tendon. So tendons are going to attach muscles to bones. Tendons are going to attach muscles to bones. Ligaments attach bones to bones. The tendons are going to be dense regular connective tissue. Finally, we have an aponeuroses. Aponeuroses is a connective tissue sheath. So such as this connective tissue sheath over the rectus abdominis. we see an epineuroses or dense irregular connective tissue. There's going to be a deep and superficial fascia. The deep fascia is surrounding the muscle tissue proper. The superficial fascia is surrounding the exterior portion. So it's going to be around the adipose tissue. This is also known as the hypodermis or the subcutaneous region. So we saw that with the skin and the integument. So this deep fascia, which is covering the actual muscle fibers themselves, is characterized as dense irregular connective tissue. The superficial fascia is going to be composed of partly adipose and areolar connective tissue. So here we have the most superficial component of the skin. Then we see the superficial fascia wrapping around with adipose and areolar tissue. Then we have the deep fascia represented by this green sheath here. And this is dense, irregular connective tissue. So some matching for our important structures here. All right, let's start with what we know first. Well, when we see deep fascia, and then we look at our answer choices. Here, it looks like E is our best. All right, sheet of dense irregular connective tissue external to the epimysium, separating the individual muscle fibers while binding groups of muscle together. Superficial fascia. This is going to be G, the areolar and adipose tissue located between the skin and the deep fascia. For epimysium, this is going to be B, the areolar connective tissue wrapping. It's delicate, providing electrical insulation, capillary support, and binding the neighboring cells together. Perimysium is going to be D, dense irregular connective tissue wrapping the fascicle, containing nerves and blood vessels. The endomysium is going to, sorry, I switched that up. The endomysium is going to be B, the areolar connective tissue wrapping the muscle fiber, right? The epimysium, that's the outermost, right? So the epimysium is going to be F, the dense irregular connective tissue wrapping a whole muscle. So flip those around. I missed that. Epimysium is the outermost, so that's going to be wrapping the whole muscle. Endomysium is the areolar connective tissue wrapping the individual muscle fiber. Next, we have a tendon. Tendon is going to be the cord-like structure of dense regular connective tissue. And finally, we have an aponeuroses, which is going to be C, the thin, flattened sheet of dense irregular connective tissue. So a normal cell is what looks like this over here on the eukaryotic cell. Normal cell is going to have one nucleus, and it's going to have all of these separate organelles that exist in the cytosol or cytoplasm of the cell. A muscle cell is going to be a little bit different. For a muscle cell, we're going to have a nucleus, but for skeletal muscle, we are multinucleated, meaning that we have 2 or more nucleuses per muscle cell. It's going to be much longer, but it's going to be more spindle-shaped than a normal eukaryotic cell, which is usually more spherical and more condensed. Here we see normal cells in terms of their binding. In a normal red blood cell, binding of oxygen happens at the 4 heme groups. So oxygen delivered in the blood is by the hemoglobin molecule. We're utilizing glucose for cellular respiration here. In the muscle, however, we have a storage protein for oxygen similar to hemoglobin. This is known as myoglobin. Myoglobin is going to be able to store 2 oxygens per molecule, whereas hemoglobin is able to store 4. So to get energy from muscle movement, we use the cellular current, which the cellular energy substrate is ATP. We can only store a finite amount of ATP at any one time. So if we start out with very intense rapid activity, it's usually only about for the first 5 seconds or so that we're able to sustain that activity with stored ATP concentrations. So after that, we have to start making it, and we have to make it quickly. So immediate is going to be the phosphate transfer. And that is going to give us a straight up supply of ATP, very minimal, but also very fast. Then we have short-term supply of ATP for maybe up to a minute, by glycolysis, which is the breakdown of glucose in the absence of oxygen. And then we're having long-term supply of ATP and the oxidative phosphorylation reaction, or the aerobic cellular respiration. This happens in the mitochondria, and we are typically going to be able to sustain our energy reserves for minutes to hours for long periods of time. Our immediate supply is dictated by our enzymes. We have the enzyme myokinase. Myokinase is going to be a kinase enzyme, and it's going to act to put things together. So here we take an inorganic phosphate from an ADP, and we're going to transfer it to another ADP so that we can regenerate ATP here. For creatine kinase, this again is going to be an enzymatic activity. In this case, we're transferring an inorganic phosphate from creatine to ADP. So instead of taking it from another ADP, we're taking it from creatine. We're moving that phosphate off onto ADP, and you have creatine left over as a byproduct. This immediate supply, this immediate phosphate transfer is only going to last about 10 seconds or so. For glycolysis, we can last a little bit longer. We have an anaerobic metabolism, which means that we can utilize glucose to be able to create energy without oxygen here. That glucose is going to be converted into 2-pyruvates. Once we have the 2-pyruvates made, we get 2 ATP released by a byproduct of that reaction. So each time that glucose circles through the process, we can potentially get 2 ATP out for each glucose that goes through. This is going to only last for a short period of time because after a while, lactic acid begins to build up. And in order to get rid of that, we have to be able to oxidatively metabolize it. So if we are exercising intensely and we reach a point of exhaustion, usually it's going to be from lactic acid buildup and not so much physiological exhaustion. So this ATP supply can last for up to a minute of exercise, and then we're going to have to start using oxidative metabolism, or the lactic acid will build up too much and there will be toxicity. For long-term ATP supply, we're going to be utilizing the aerobic oxidative transport, oxidative phosphorylation chain. So this requires that we get oxygen in and that we take that oxygen into the mitochondria. There are 3 stages: intermediate, Krebs, and oxidative phosphorylation. Our main storage form of calories and energy in the body is adipose fat. So triglycerides are also able to be used to produce ATP if that is all that is available. We are going to get significantly more ATP from triglycerides than we do from other molecules, mostly because of its fatty acid chain. So when we are in an exercise state, we are then going to owe what is called an oxygen debt. The oxygen debt is the amount of oxygen that we need to get back in after exercise to reestablish what we were before that. So the additional oxygen is going to be there to replace the oxygen on the hemoglobin and myoglobin, replace glycogen storage, replace ATP, and replace creatine phosphate. So let's look at important structures of the muscle cell. First, we have the sarcolemma. Right? Sarcolemma is the storage place for calcium. So it's going to be where calcium is stored. T-tubules. T-tubules are going to be the modified smooth endoplasmic reticulum that is going to contain calcium pumps to pump calcium into the terminal cisternae. Next, we have the sarcoplasmic reticulum. Then we have the terminal cisternae. Then we have the triad. Now let's work back through those. The triad is going to be G, location where 2 terminal cisternae meet at a T-tubule. Sarcoplasmic reticulum is going to be basically what we would call the plasma membrane of the muscle cell. So we see that here. See the triad, just went over that. Calmodulin. Calmodulin is there to modulate calcium. Calsequestrin is going to be there to sequester calcium. Then we have two receptors, a ryanodine receptor and a dihydropyridine receptor. Ryanodine receptor is going to be inside of the T tubules, and the DHP or dihydropyridine receptor is going to end up being the calcium release channel. So here's our normal cell. We have ion movement back and forth. Here's our muscle cell. All right, let's go ahead and watch a short tutorial on that.
It was time to get my license in teaching formally. I've been teaching informally for a long time.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms.
All right, so that's what that looks like. The terminal cisternae, sarcoplasmic reticulum, and the T-tubule structure. So back to our PowerPoint here. We have the flow of ions through transverse tubules out from the sarcoplasmic reticulum into the terminal cisternae. Here we have a T-tubule representation. T-tubules, as stated, are going to be small invaginations in the plasma membrane that allow for ion conduction down and in. So they contain a lot of different channels, one of which is going to be the voltage-gated ion channel, allowing for electrical signals to come down. So they have a non-specific channel for electrical conduction going down in between. More receptors. Again, we have the dihydropyridine receptor. This is going to be a voltage-gated, so they're voltage-gated calcium channels. It's going to be bound to ryanodine receptors in the sarcoplasmic reticulum. So here's our sarcoplasmic reticulum. Here's our smooth endoplasmic reticulum. You can see that there's a large difference, and don't forget that difference. All right, so here's an example of a pump across that membrane. We have a calcium pump and a calcium channel. We have calmodulin and calsequestrin holding on to calcium ions within the terminal cisternae. Once the pump is stimulated, it will release that calcium atom out into the sarcoplasmic space. So if you read a textbook, especially something like the Whole Anatomy and Physiology, it will state that the calcium channels are going to be just known as that, calcium channels, but they are really ryanodine receptors. They are a mechanical receptor, which means that they react to a change on the DHP receptor. So when there's a millivoltage change on the DHP, that means that there's going to be some sort of a change also at the ryanodine receptor, because we are stimulating it to move away from or closer to the site of attachment. All right, so this is the voltage-gated calcium channels. All right, we have the dihydropyridine receptors here. See the two-tubule structure as a whole here. You can see some leak channels for potassium and sodium here. And there's our neuromuscular junction. All right, so some of our muscle cells are going to be slightly unique. In a normal cell, we're going to have some mitochondria, but lots of intercellular fluid. In a muscle cell, we're going to have full contractile proteins. So it's not going to be nearly as much open space, and it's going to be much more organized. All right, so here's an example of a myofibril that's surrounded by an endoplasmic reticulum. You can see that each myofibril is surrounded by an ER, and each ER is a place that you can have calcium binding and then get sequestration of it out into the muscle fiber tissue. All right, myofibrils are 80% of the volume of a cell. They can range anywhere from the hundreds to thousands as terms of population. There are two filaments that we see inside that are contractile of nature. We see the thick filament, which is also known as the myosin filament. It has a head group, an actin binding site, and an ATP binding site. Thin filaments are known as actin. Actin is going to be covered up twice, once by tropomyosin and then the other time by calcium binding. So if we have a low amount of blood calcium, that means the tropomyosin is not going to be stimulated as nearly as often or as quickly to generate the same amount of force. So let's do this chart really quick on myofilaments. So I'll start from the bottom. It says myosin binding site. So the myosin binding site is going to be the area on the G-actin where the myosin head can be hit, and that is letter E. Troponin. Let's see if I can find it. Okay. So troponin is going to be F, short, thin, twisted protein in a string-like formation. Tropomyosin, right? Excuse me. Tropomyosin is actually going to be the filamentous actin string of G-actin. Troponin is going to end up being D, the ball-like protein containing calcium binding sites and attaching to tropomyosin. Next is the myosin head. This is H, the head portion of myosin, aka the myosin ATPase. And that head portion is going to be the myosin ATPase, which is enzymatic activity. So head portion of the myosin protein has enzymatic activity. And we have thin filaments. This is J, the two strands of F-actin, troponin, and tropomyosin. We have thick filaments, right? Thick filaments are B, bundles of myosin protein. So this is the structure of the skeletal muscle. You can see an individual nucleus there, one there. All right, we see all the fascicles of muscle tissue running down, and we can pull back the periosteum to reveal all this open space. Right, next is the functional unit of the muscle. Functional unit of the muscle is known as the sarcomere. Sarcomere runs from what is called a Z disc to a Z disc. So we have a Z disc or Z line here. We have a Z disc or Z line over here. And this entirety of this structure is known as what we just went over, which is the sarcomere. So portions of the sarcomere. One is the thin filaments. This is the F and G actin. All right, we also have the troponin and the tropomyosin up here. We have the myosin heads. They're all cocked on the thick filament, ready to go. All right, next is what we call connectin. Connectin, which is also known as titin, is the largest protein in the body. It's going to lick the thick filaments of the myosin to the Z disc. Then, as mentioned, we have our thin filaments, which is the F and G actin. The sarcomere structure, we use it based on zones. So first off, we start at the M line, which is the middle line of the sarcomere. Next, we have the H zone. During relaxation, the H zone only contains thick filaments. Then we have the A band. At relaxation, the A band is from one end of the thick filament to the other. So it contains about half thin filaments and all full filaments at any one time. Next is the I band, right? So the I band is where you have communication with the other cell. So this is the distance between the end of the thick filament and the beginning of the thick filament on the next muscle. All right, so here's a sarcomere shortening example. This sarcomere up here is relaxed. We have all of our zones present. Everything is open and everything is able to be seen. Below that here, we have a fully contracted sarcomere. With the fully contracted sarcomere, now the Z discs have been pulled closer towards one another. The myosin heads have been cocking and pulling, like Fallout Boy, the actin proteins in towards the M line or towards the center to where now they are overlapping. So there's no more H zone. The H zone is gone. The I band is virtually gone. And now we have left over an A band in the center. Still going from one end of the thick filament to the other end of the thick filament, we have that A band in the center surrounding it. So we go from the relaxed stage here, where all of the things are similar, to the contracted stage here, where we have compression and also disappearance on some things. And so to the right, this is a microscopic image of that. We see the H zone right here, the M line in the middle of the H zone. We can see the A band, right, one end of thick filament to the other end of thick filament. And we can see the I band, one end of thick filament to the end of the thick filament in the adjacent muscle cell. So some important things to note here as far as important structures of the sarcomere. We have the sarcomere itself, all right? Sarcomere itself. Let's see what I can answer first here. I never go through these questions before I do these, so I'm answering them real time, which gives everyone a real nice way to poke fun at me, which is fine. I want you to laugh at my expense. I bring joy in many ways. All right, so first, I'm going to go with H. H is connectin, right? That's the specialized proteins appearing perpendicular to the myofilaments. Then we have the thin filament. Thin filament, that's going to be the filament made of two strands of F-actin, tropomyosin, and troponin. We have the thick filament. Thick filaments are going to be the bundles of 200 to 500 myosin proteins. The M line is going to be I, central region of the sarcomere that contains the entire thick filament, appears dark in the middle. We see striations here. Striations, not quite sure which one they're looking for. I'll come back to that. H zone is A, this is the most central part of the A band. contains only thick filaments and disappears upon muscle contraction. We have the I band, which is going to extend outward in both directions from the Z disc, containing only thin filaments, disappearing upon contraction. Next, we already did M line, we did H zone. Sarcomere as a whole. Sarcomere as a whole, I'm going to go with C, the repeating cylindrical units of contractile protein organization. For striations, I'm going to go with J. That's how sarcomeres at rest are viewed under the microscope. And I think that completes this. So I think we did sarcomere, Z disc, I band, A band. I don't think I missed any. If I did, shame on me. All right, so this is what a microscopic view is going to look like of that. Again, we have the sarcomere, which goes from Z disc to Z disc. We're going to have that H zone in the middle, containing only the myosin. We have the M line, which is the exact equatorial middle line of the sarcomere. We see the A band, which goes from one end of the thick filament to the other on the other side of the same sarcomere. And we have the I band, which is going to contain the area between the end of a thick filament on one sarcomere to the beginning of a thick filament on an adjacent sarcomere. So here's a definition of those. This is the Z line here. Then we see just actin filaments here. Then we get into the A band where we see overlap of thick and thin filaments. And then we get to the center where we have the H zone where it's just the thick filaments. And then we see the middle line, which is the M line, which is the proteins. All right, everyone, that is organization of skeletal muscle and what the units of skeletal muscle look like. Thank you for joining me, and I'll see you at the next video.